Hello everyone and welcome to today's webinar, Prepared for the Next Pandemic, Utilizing New Production Capacities for Streamlined Vaccine Manufacturing. I'm Tim Wright, editor of Contract Pharma, and our webcast today is being sponsored by IDT Biologica. They're a leading CDMO providing end-to-end services in development and manufacturing of viral vaccines, cell and gene therapy products, and aseptic fill finish for other biologics and sterile injectables. The company has GMP sites located in Germany and in Maryland in the U.S. for clinical and commercial manufacturing, and it maintains a clean track record of successful inspections and approvals from major regulatory agencies, including FDA, EMA, and ANVISA, delivering commercial programs for over 3 decades. You can visit them online at idt-biologica.com or simply by clicking their logo at the top of the page. We have several speakers today, including Daniel Miner, Head of Manufacturing and Senior Production Manager at IDT Biologica. Joining him is Dakota Chenoweth, IDT's Team Lead for DSP Technologies Process Development. And we'll also hear from Daniel Heineck, one of IDT Biologica's scientists for upstream process development. Over the next 45 minutes or so, you're going to learn about the critical aspects of pandemic preparedness, with our speakers focusing specifically on vaccine development and production, and the methodology IDT used to establish a production process potentially capable of producing enough vaccines to ensure rapid distribution to the population in the event of another pandemic. And along the way, we'll also get a close look at IDT Biologica's newest state-of-the-art production facility. We will be accepting questions throughout the talk. You can send those in using the box at the bottom of your screen, and questions will be answered after the presentation in a brief Q&A. To enhance your viewing experience, feel free to customize your window display. You can move the windows around by clicking the top header of each window and dragging them around. You can also activate the closed caption function by clicking on the CC button in your media player. And you can share your reactions to today's content with the emojis provided. And today's speakers will be able to see your reactions in real time. And lastly, if you're experiencing any technical difficulties, you can type those into the question box as well, and someone on the back end here will help you out. So at this point, I will turn it over to Daniel Miner.
Yeah, hello everybody. Thank you, Tim, for the kind introduction. Today we want to have a closer look to the pandemic readiness of IDT, looking to new production capacities as well as the development of new processes at IDT. Yeah, as Tim already said, IDT Biologica is a globally operating biopharmaceutical CDMO that handles with vaccines, gene and immune therapeutics, and fill finish of biologics and other sterile injectables. Our mission is to accelerate the development and the manufacturing for our clients for therapeutics and prophylactic products. We're handling with healthcare and treating of diseases with worldwide impact. We have the whole value chain in-house, from process development to commercial manufacturing, as well as quality control and analytics. IDT Biologica is a private-owned company held by the Glocke family, Carsten Glocke and Stefan Glocke, and at the moment our CEO, Dr. Ulrich Wallay. We have operations in Germany and in the US, and a history of over more than 100 years within development and manufacturing of vaccines. And in the last 30 years, we also made our experience in commercial filling. And over the last years, we had a lot of invests in some over more than 600 million euros. At the moment, we have around 1,700 employees, and we are approved by the EMA, the FDA, and the ANVISA. As a leading global CDMO, we can support our customers with the complete pharmaceutical life cycle, from preclinical to commercial production for vaccines and cell and gene therapeutics. For biological and sterile injectables, we can support with late-stage development up to commercial production. IDT Biologica is mainly located in Dessau-Rosslau at the Pharma Park. Here we see a short overview of the pharma park in Dessau-Roßlau. On the right side of the pharma park, we have mainly our pharmaceutical activities, and on the left corner here, we have our vaccine activities. So we will have a closer look in the next slides to our vaccine activities over the site of the campus. As already said, we had some big invests in the last year at the IDT. Some examples are the filling facility we opened up in 2015, our -80-degree storage, and the multi-packaging line we implemented in 2016, and our state-of-the-art high-speed isolator filling line and the second filling line we finalized in 2017 and 2022, and as well to DS manufacturing buildings. One we implemented in 2019 and one we implemented last year. One of our latest invests is a high-speed filling line, suitable for 2R, 6R, and 10R vials. Here we can realize batch sizes up to 500,000 vials with a filling speed of up to 32,000 vials per hour. Looking to clinical activities, we also have capacities available where we can fill smaller batch sizes. In general, we have 11 filling lines at IDT available, where we can support small batch sizes and, for example, batch sizes of more than 100,000 vials for lyo processes. Now we will have a closer look to the construction of a new drug substance facility we had over the last years at the IDT campus in Dessau-Roßlau. The expansion of the DS capacity at IDT Biologica we will want to have a closer look to starts in April 21. Here we focused ourselves on the technical functionality on the building, and within every big invest there was a great need to save cost and time. So here we had a timeline of less than 25 months to build up the whole thing and to be technically ready. On the picture on the right side, you can see that we built up the new facility direct next to already existing drug substance facility. That made us able to connect to already existing media systems like Wi-Fi, clean steam, pressured air, and so on, so that we can save the supply level and to have a short way to implement systems like Wi-Fi, clean steam, and so on. We realized the construction of the whole building as a room-in-room concept that made us able to divide the whole construction in two phases, so that we were able to start the qualification in the part of the building we already finished while we were running under construction in the second phase. So that made us able to speed up the whole thing with the qualification as well as in the building of the facility. As I already said, we start with the construction in April 21 and we end with the technical construction in December 2022. We received the GMP readiness mid of 2023 from the German authorities, and in sum we realized a clean room area of 1,700 square meters with 8 process rooms where we can run the later on processes in the new facility. So now we will have a closer look to the new drug substance capacity that we realized at the IDT campus. On the picture, you see the layout of the production area. Here, where we realized 8 process rooms, as well as personnel and material locks. We have introduced cold rooms for 2 to 8 degrees storage and minus 20 degree storage, and possibilities for general storage that is necessary for the later on production. In this slide, we see our production rooms in blue, given the cell upstream suites, and given in green the suites where we do the upstream and the downstream of our active materials. We have highly flexible process rooms. They are nearly completely empty once we start with a new process. Our cell upstream rooms are exclusively for cell culture, so no active materials like viruses, plasmids, or so on were handled in that rooms. And to keep the rooms clean and to have a proper cell cultivation, we have a slightly overpressure in that rooms. The rooms for the manipulation of active materials are not strictly dedicated to the upstream or to downstream processes. So, as already said, these rooms, all of our rooms, are highly flexible. And for the environment of handling active materials, we have a slightly under pressure in the rooms to ensure that we don't have an exit of active materials from the room. Within our facility, we need to handle open process steps. Therefore, we have clean rooms with Class C available and a Class A biosafety cabinet. These rooms are given in green in the picture. Closed processes will take place in the Class C environment, given in blue in the picture. The personnel locks and the material locks within the facility are given here in green. So here we separated the material and the personnel flow. For our personnel flow, we have a separate entry and a separate exit of the personnel from the process rooms, and we separated the material and product flow from the waste flow so that we have a proper way out of the process room for products as well as for the waste. Here we see 2 examples for the personnel lock. Once we enter the cell preparation and once we enter the virus production. Here the personnel lock is the pressure trap, so that we ensure that we don't have an inflow to the process room within the cell culture, and to ensure that we don't have an exit of materials from the virus production. As you can see here, we separated the way in to the process room and the way out of the process room. The first, or the direct contact room to the process room, belongs to the containment of the room and will be disinfected and decontaminated with the room. So this is why we located the pressure trap in that room. It is the same for the material lock. The material lock, we decided to have a one-room lock, so that this room is also the pressure trap to ensure that we don't have an inflow to the cell culture room, or that we don't have an exit from the virus production room. So also this room belongs to the containment of the process room. As I already said, we separated the waste flow, especially the waste flow of our solid infectious materials from the product and the material flow. So all of our solid waste will be collected within a waste corridor in decontamination autoclave at the end of the corridor, so that we can ensure that no infectious material will leave the facility, and to have a proper waste management. Disinfection and decontamination of our process rooms will be done by hydrogen peroxide gassing. Within that facility, we'll have a closer look to it. It is a completely automated process. Here we need gas-tight rooms, and we were working with a hydrogen peroxide concentration of 350 to 400 ppm. At the beginning, we need to adjust to a humidity of lower than 45%. The whole process takes around 16 hours, where the degassing of the room takes the much time within that process. And after we went down to an acceptable level of hydrogen peroxide, less than 1 ppm, we can enter the room and release that room again. Within our facility, we have different capabilities to handle cells. One capability is to handle adherent cells. Here we have CO2 incubators available to incubate T-flask or cell stacks, as well as the manipulation of CF40s. Here we were able to handle 16 CF40s in parallel. On the pictures, you can see on a CF40 manipulator that brings the CF40 racks in a position where you can fill or empty them and do your harvest, anything that is necessary within your processes. And on the right picture, you see CO2 incubators built up to handle these CF40 racks, where we have 4 CF40s in one rack. Within the facility, we are also able to handle suspension cells. For suspension cells, we also have CO2 incubators available, where we can handle a shaking flask with a scale of 100 milliliters up to 5 liters. And above that scale, we have bioreactors available for the cell upstream, 50-liter bioreactor, 200-liter bioreactor, and a final scale of 2,000 liters. So here we can handle suspension cells from a milliliter scale up to a 2 cubic meter scale. Our downstream capabilities are filtration in any kind you can imagine: depth filtration, membrane filtration, sterile filtration. We also have TFF systems available, and over the last years we get in touch with chromatography in any kind, ion exchange, size exclusion, membrane chromatography. So we are well prepared to any kind of downstream activities you can imagine here. To summarize the expansion of our DS capacity, we have built up 8 suites with a biosafety level of up to 2. We were able to handle 6 bioreactors with a scale of up to 2,000 liters to be able to perform 160 batches over a year. We were completely flexible within our upstream, downstream technologies for adherent or suspension cells. If we need to adapt our suites, we will do that for our clients. We can also handle iCellus 500 within the facility to deal with adherent cells. So in general, we were able to handle cell factories up to 16 within a batch. handled by a robot. We can handle isocellus processes and stir tank reactor processes up to 2,000 liters. So thank you, everyone. This was a short overview about the new DS capacities at IDT Biologica in Dessau-Rosslau. Now I will hand over to Dakota, and she will start to go more in detail to the pandemic preparedness of IDT Biologica.
Thank you, Daniel Mineur, for your introduction into IDT's latest investments and introducing our new state-of-the-art production capacities. I'd like to share with you IDT Biologica's road to pandemic preparedness. IDT Biologica is an official partner in the Pandemic Preparedness Program, where contracts from the Center of Pandemic Vaccines and Therapeutics in Germany, also known as CPI, were signed with multiple German pharmaceutical companies, as seen in this picture above. Contracts ensure manufacturing capacity is guaranteed for rapid production and distribution in Germany and the EU in the event of a future pandemic. As of mid-2023, IDT Biologica was the first company to achieve the pandemic preparedness status. But first, what is this pandemic preparedness that we are talking about? Let me introduce the Pandemic Preparedness Plan. Our aim was to develop a generic 2,000-liter scale adenoviral suspension process to demonstrate IDT's pandemic preparedness. The expression system used was the HEC 293 suspension cell line, which is commonly used for manufacturing adenovirus based on research of adenovirus platforms. We decided to use serotype 5 as our adenovirus model virus. Our platform is a model process, and therefore the adenoviral serotype can be substituted through other serotypes if needed. For the process design, multiple publications were considered, with the published ChAdOx manufacturing platform being the most significant for the initial process design and planning. We strived to design a generic approach for the adenoviral platform to be processed in our new production facility, also based on the large production systems that were already implemented there, like Daniel just shared. Lastly, a large focus was put on using EU supplier materials that are produced in the European Union to prevent bottlenecks from occurring and to limit long shipping times. This was a key element of the pandemic preparedness that was incorporated into the process design. I will give you a detailed inside look into this shortly. But first, I would like to present our strategy map. To become prepared for the next pandemic was no small feat, and many different departments with numerous colleagues worked closely together over a year to achieve this pandemic preparedness. In the center of our strategy map is our generic process design, which includes a tech transfer database where all process details and parameters are specifically listed for the scale-up and transfer from development into production scale, as well as the risk assessments as in the FMEA and the APMP, also known as Analytical Project Master Plan, which all play an important role in having a robust process design. As you can see, to achieve a generic process design, many departments were incorporated and worked hand in hand. Today we will highlight the collaboration between the process development department and the technical operation at the GMP facility, which my colleague Daniel Mineur already spoke about. Before we dive deeper into the process development and process design, I'd like to say a few words about these other important departments. Procurement played a very important role in the EU supplier concept and planning all the materials that would be needed for such a large-scale production, as well as the countless contracts that needed to be established between these suppliers and the IDT. Human resources was a key player in supplying highly qualified staff to be able to perform these throughput difficult processes. Our team is just that, highly qualified in numerous processes, having specialized upstream processing and downstream processing teams that can handle even 2,000-liter manufacturing with GMP quality and in well-coordinated 3-shift systems to allow for continuous production of drug substance material. Validation, quality control, and supply chain were also important to establish a qualified product in supplying risk assessment, specific process requirements, and the APMP. Our department, Process Development, designed the generic process, but worked very closely with Daniel Munoz's department, Technical Operations at the GMP facility, to ensure that the tech transfer and all the various parameters could be seamlessly transferred, and that the chosen materials and systems could be combined for the optimum process. With a new platform, new analytical methods needed to be established, which was also coordinated in close conversations with our colleagues at IDT's analytical development team. For our development approach, 3 main factors contributed to our process design: the production facility design and capacity, the process expertise that IDT has procured, and the EU supplier concept. First, we checked the available systems in our new production facility and all the parameters, pump capacities, and volumes that could be used, as well as the facility design, as the cell culture material needed to be transferred from one suite into the next where the stirred tank bioreactors were located. This material transfer also occurred after lysis, when the material was handed over from the upstream team to the downstream team for purification. Lab organization was also important to keep in mind, as the space is limited, especially in the downstream processing suite, where a lot of storage is needed for the large volumes of buffer for the chromatography steps. This was all planned out in precision, as well as the waste management. Using IDT's waste management system, the waste could continuously be pumped out of the production room and be inactivated. Secondly, IDT's process portfolio and accumulated know-how from many other processes helped shape the process design, and especially handling and workflows. Support from external partners ensured our scaling was accurate between small bioreactor systems and smooth transfer to the large-scale systems, especially when these were from different companies and had different setups. And as mentioned earlier, literature data and analysis was also incorporated for additional cell line characterization and information on adenoviral processes. Lastly, the EU supplier concept, which I have already mentioned, played a large role in determining which materials could be used. Companies that had a large range of scalable materials for both the upstream and downstream processing were researched and evaluated. Buffers also had to follow this concept. Multiple contracts were established with these suppliers to ensure ready materials in case of another pandemic. Let me describe our adenoviral manufacturing platform design to you on the next slide. This is an overview of the adenoviral manufacturing platform strategy that was designed. The HEC 293 suspension cell line is seeded and expanded in shaker flasks before it is then transferred into a stirred tank bioreactor. Since our goal was to produce 2,000 liters of virus harvest, it was no option to expand the cells only using shaker flasks due to the sheer amount of cells needed. Therefore, we have implemented a cultivation strategy where we utilize the smaller stirred tank bioreactor scales that are available. The cells were passaged from shaker flasks into an Allegro 50 and then further into an Allegro 200 bioreactor. The final passage could then be performed in the Allegro 2000 using a fed batch approach for virus infection and final cell lysis. An optimized MOI for efficient adenoviral manufacturing was used to infect the cells. After the correct TOH had been reached, cell lysis combined with nuclease treatment for the DNA digestion occurred in the Allegro 2000. Let me highlight for you that we can, of course, do any manufacturing batch size up to 2,000 liters, so that clinical trial material manufacturing from phases 1 to 3, where usually less material is needed, is also possible. After the lysis and DNA digestion step, the harvest takes place. The material is then pumped through the wall port into the DSP processing room, directly through the first clarification filter and then through the second clarification filters, where it is then stored in a 2,500-liter LevMix storage container for further processing. The next purification step is the anion exchange chromatography that is performed using a membrane-based chromatography approach in multiple cycles to be able to process this large volume. After the AX chromatography, the material is concentrated and diafiltrated via tangential flow filtration on our large ACTA ReadyFlux XL system. Both hollow fiber and cassettes were tested and are both part of the strategy depending on which materials are more readily available. Thereafter, a formulation is implemented before the final bioburden reduction filtration is performed. Afterwards, the BDS is stored at greater than negative 65 degrees Celsius for further drug product use. Drug product will then formulate and fill and finish the BDS for final dosaging. This strategy was designed in small scale to be transferred into large scale. My colleague Daniel Hünig will tell you more about this scale-up process.
Thank you, Dakota, very much for introducing us into the Pandemic Preparedness Program and our adenoviral large-scale process that we have come up with. Now, as Dakota has mentioned, there were several considerations that influenced our process design, and what I would like to take you through now are our technical approaches and our transfer procedure. First, let's take a look at our tools that we have at our disposal. For the upstream process development, we have technologies such as the Ember 250 high throughput, a system for small-scale bioreactor approaches up to 250 milliliters, allowing to conduct screening experiments and statistical DOE approaches. Having then determined a set of parameters, we can scale the process up into our Cyvario system. We can use 3-liter vessels for the confirmation of our pre-established processes and further scale it up into 10 and 40-liter vessels as a proof of concept. We are also using these systems to provide material for the downstream process development, where usually higher volumes are needed. As you can see, we are mainly using ACTA systems for both chromatography applications and tangential flow filtration, where you can use various sizes and types of columns and hollow fibers. By changing the column size or adding additional chromatography cycles, anything from 1.5 liters up to 20 liters can be processed on our ACTA Pure chromatography system. Depending on the process, larger scale tangential flow filtration can also be performed with our Repligen cross-flow system, but that was not the case in this process. And then finally, we have the commercial manufacturing scale up to 2,000 liters that we have aimed for and that is needed during a pandemic. And here I just want to give you some impressions from our development department. We can see our Ember 250 HT system, where we have conducted screening experiments and up to 12 small bioreactors per experiment, so we could screen for more parameters or specific parameter combinations simultaneously. And this is a big advantage to usual benchtop reactors that mostly take up higher volumes and are limited to the number of reactors and therefore expensive and time-consuming. Here we can also see the Cyvario system where we have conducted our scale-up and confirmation as a proof of concept. You can also see our ACTA Pure for chromatography and ACTA Crossflow for tangential flow filtration applications. As mentioned before, we can process with different sizes and types of columns and hollow fibers. Just briefly, that you can get an impression of our practical development work. I would also like to point out that scale-down procedures are also possible, for example, to further optimize already existing processes. Now, let's talk about how we have ensured the smooth transfer from our development scale into production. After our small-scale process was established, we have conducted confirmation runs in the scale of 3 liters. At that time, the responsibility was mainly on the side of the development team to perform the investigation. The task of the production team was to give some guidance in order to improve the developed process later on. Following this, we have performed an engineering run in the final production scale of 2000 liters with the final technology, namely the Allegro systems. As mentioned before, we have performed scale-ups into 10 and 40 liters as a proof of concept, but the reference process was the 3-liter scale process. And at that stage, the responsibilities were equally distributed amongst both teams, which was the key to a smooth transfer. It looked a little like this. The development team has transferred the process in cooperation with the production team by being present on site, giving advice, and discussing the procedure with the production personnel. Finally, qualification runs, which later would state the usual manufacturing process, have been carried out, where the production team was completely responsible for the manufacturing and the development team had only a supportive role if questions arose or helping with data analysis. I believe that some people working in this very specific field of application may find that when transferring processes, it is often the case that parameters and technical procedures are simply handed over in theory from one team to another, somehow cutting the tech transfer. But as you can see here, in our case, there was no hard cut in the transfer procedure. By the 2 departments working closely together, we have accomplished a smooth transfer. It goes along with our first time right philosophy. Okay, now I want to give you some technical important factors that are necessary to consider for a seamless scale-up and transfer. So, first of all, of course, critical process parameters have to be understood, like gassing rates or the gassing regime in a bioreactor. Sometimes the practical scale-up is not possible according to the calculated theory, due to the scale itself or the system change, like a different bioreactor. In our case, the geometry changed from a cylindrical geometry in development to cubical in production. So know your process, adapt the parameters in such a way that you can achieve the same biological, chemical, and physical properties. Another thing to consider is comparability of equipment between development and production. And this is not about the obvious equipment such as bioreactors or filtration devices. This is more about the equipment that is additional to the process, such as analytical devices, cell counters, for example, and cultivation equipment. If they are not the same, which often might be the case, compare the data and adapt the parameters so that, bottom line, the same process takes place. Next on is an understanding of flow limitations. Check beforehand if the developed flow rates go along with the system pump capacities and tubing lengths. If not, develop a strategy with which the fluid can be transferred into the target system without harming the process and product quality. And the last point is that one has to consider there are only certain sizes and formats of materials for large-scale production, filters and columns, for instance. The process has to be designed in such a way that it can fit into these sizes and formats. The important lesson you can learn here is that scale-ups and tech transfers are separate things that mostly do not go along with each other, and compromises have to be found between them. While scale-ups are basically mathematical calculations, tech transfer describes the procedure of transferring a process from one system to another, where the limitations are different, especially when working with systems from different manufacturers and suppliers. And that is not only restricted to devices and materials, but also to locations and when we talk about scale-up, to the increased volumes themselves that come along with variances and challenges. And these are important aspects to consider at early stages of the development, where close collaboration between teams and departments are necessary to tailor a suitable process. To bring this presentation to an end now, here are the today's learnings that you can take home. We have presented to you our brand new production facility for GMP-compliant manufacturing. With the state-of-the-art equipment, we can operate numerous platform technologies. Furthermore, you have learned about our pandemic preparedness program, our development approach on how we have designed a large-scale process to demonstrate our readiness for a possible next pandemic. And last but not least, you have gained deep insights into our development and our procedure to a smooth and successful technology transfer. And with that, I would like to thank Tim and the team of Contract Pharma for the organization and hosting this webinar, my colleagues Dakota Chenoweth and Daniel Mineur for making this presentation with me, and finally you, the audience, for your attention, your interest, and for being with us today. Thank you very much.
Okay, great. Thank you so much to our speakers, Dakota, Daniel M., and Daniel H., for a great presentation today. So we do have time to answer some of your questions now. If you do have a question, weren't able to get it in during the talk, now's the time you want to go ahead and type it into the question box at the bottom of your screen there. Also, before we get to the Q&A, just want to make a note that while a PDF of today's PowerPoint is not available to the audience, IDT has made several assets available for you to download, and you can do so by hitting the Resources tab on your screen there. Okay, so let's get into the Q&A here. This attendee would like to know, in what other platforms does IDT have know-how?
Okay, thank you, Tim, for this question. Very excellent question. So basically, we do anything that has to do with the production of viruses, either if it's for vaccines, for cancer, or for gene therapies. And yeah, we basically operate suspension technologies, as we have presented here right now, and as also Daniel Mineur, my colleague, has mentioned beforehand, we also do operate adherent cell culture technologies. So, for example, the iCellus technology, not only in the production where we use the iCellus 500, but also here in our development department, we are using the iCellus Nano technology, and we can also work with universals, fixed beds. So we have a quite broad range when it comes to cell cultures and suspensions, and yeah, basically the whole upstream production. Also, static systems such as cell stacks are also at our disposal.
Right. Okay. Thank you.
Yeah.
I'm sorry. Do you want to add anything else?
Yeah, I just wanted to add that we also operate in various downstream technologies such as chromatography systems, different types of chromatography systems, and also different types of tangential flow filtration. Yeah. So yeah, basically that's it. Thank you.
Okay, perfect. Thanks so much. All right, moving along here. This attendee would like to know, how do you handle waste air and heat in a clean room from CO2 incubators?
Maybe I will jump into that question. Thanks for that question. So here we have CO2 incubators in our rooms, up to 6 here within. We have an active air supply within the rooms and active control of our temperatures in our rooms. So waste air and heat from our CO2 incubators is not an issue for us because of the fresh air supply to our rooms.
Okay, great. Thank you, Daniel.
You're welcome.
This attendee asks, she says, I might have missed, but back on slide 6, she says, what does CZ vial line stand for?
I can answer that.
Okay.
Yeah, so CZ is for the crystal zenith vials, which are favored by clients now, especially in the AAV production. So yeah, that's what it stands for.
Okay, great. Thanks, Dakota. All right, moving along here. This attendee would like to know, how do you segregate different customers' materials?
Okay, I will jump in here. If I understand that question right, or there are two possible answers for that question. Once we're talking about the materials that needs to be used for the processes into our process rooms, here we have dedicated SAP recipes where the used or the necessary materials and medias and buffers and so on are strictly dedicated to the single processes. So with the SAP as a background for our material logistic, we separate the single materials. If the question means how to separate products or intermediates, here we separate the materials while we only have one product in one room at one time. So we don't handle or manipulate two products in the same room at one time. Hope
Okay,
this is sufficient.
Okay, thanks, Daniel. Let's see. I think we're going to take one more question before we wrap things up here, if nobody else has any other questions. This attendee would like to know, can IDT also change the biological safety level for new product up to BSL-3? And second part of that question is, and so production with BSL-3 material is in drug substance buildings possible?
Yes, it is in general possible. Takes some effort to upgrade the whole facility up to BSL-3, but in general, it's possible, yes.
Okay, great. Thanks, Daniel. All right, so if there are no other questions, that's going to bring us to the conclusion of today's session. Thank you again, Dakota and Daniel M. and Daniel H. for a really informative and insightful talk there. Thank you again to today's sponsor, IDT Biologica. Again, be sure to visit them at idt-biologica.com or by clicking their logo on your screen there. And thank you very much to everyone out there in the audience for your attendance and participation today. Lastly, this session will be archived for 1 year on our website, contractpharma.com, and you'll be receiving an email from us shortly when the session is available for on-demand viewing. Thanks so much for joining us today, and have a great rest of your day, everybody. Take care.